Those who have not subscribed to the channel, just click the subscribe button. Close to that, you can see a bell icon. Click it, you will get the regular updates. To see the portions, you have to check the playlist. In the playlist, you can see each topic class-wise. Just by clicking that, you can see all the part in that lesson.
Hello, welcome to my channel. So today we are going to start our second lesson, that is acid, bases and salt. So if you have any doubt, you can ask in the comment section. So it's a very interesting lesson, that is acid, bases and salt. Acids or bases, so we are seeing it in a daily life. Have you ever thought that you are taking in one form or other form the acids or bases? Have you ever think of that? Okay, I will just ask you, we all take orange or apple or grape or guava, any of these fruits, or tomato, or we take tamarind in our curries and all, we are using tamarind. In all these, acids are present. In all these, acids are present. Different, even our milk, curd, all contains acids. So we are using things which, or we can see that we are daily taking acids in one form or the other form. And the second one, bases. We know cake, bread or cookies. In many of these, we are using bases like sodium carbonate or sodium bicarbonate. All these are bases. So we can say that in our daily life, we are seeing acids or bases. So today we are going to learn about the acids and bases. Now the next question, how we can identify a compound consists of an acid, or if you are given a two test tube in which one is an acid and one is a base. Your teacher asks you to identify which is an acid, which is a base. In the earlier class, we have learned that acids are having a sour taste or the bases are having a bitter taste. We can't taste these acids which are used in our laboratory by tasting it. Many of the acids and bases we use in our laboratory are poisonous. So we can't taste it. So what we have to do? We need an indicator or something which shows that this is an acid or this is a base. So such substances which indicates or which shows that which is an acid or a base are called as indicators. What we call it as? We call it as indicators, or the substances which make us understand that this is an acid, this is a base. Such are called as indicators. So an indicator is a substance which makes us to identify whether it is an acid or a base. So indicators are of different types. The small class onwards, we have learned about litmus paper or litmus solution which we use to identify an acid or a base. But there are different types of indicators. So indicators can be broadly classified into many types are there. In our textbook, we are just learning only some. So indicators can be broadly classified into natural indicators, synthetic indicators, and next one is olfactory indicators. So these indicators can be classified into natural, olfactory and synthetic. From the word itself, it is clear that first we will go with the natural indicators. Natural means which we get from the nature. We are only discussing some, not more, only some natural indicators. First one is litmus. We know that we have litmus paper. What all color you have seen? It is one in blue color and the other one is in red color. So how we will identify an acid and a base by using a litmus paper? A litmus paper, when we dip a litmus paper in an acid solution, we have one bottle. We don't know whether it is an acid or a base. We take both the litmus paper, red and blue. First I will dip the red litmus paper in the solution. If it does not change into any color, when the red litmus paper is not changing into any color, then I have a guess, it may be an acid. Then I will take the blue litmus paper and just dip in the same solution. Then I can see that the blue litmus changes into red. That means that solution is an acid. So we can identify an acid by the color change of blue litmus into red, then that is an acid. And if the red litmus changes into blue, then we can call it as a base. So this litmus paper is a natural indicator, because this litmus paper we are making, or the litmus solution we are making from a plant, and it is lichen plant. We are getting that from a lichen plant. So it is a natural indicator. It's a natural indicator because we are getting it from a plant called lichen, and we are making from that, and we are getting the litmus solution or litmus paper. So litmus is a natural indicator which shows that if a litmus paper color changes from blue to red, then it is an acid. We can easily think that red means danger. So acid, always acid is more danger. So we can say that if a blue litmus changes into red, then it is acid. And if a red litmus changes into blue, one call it as base. If a red litmus changes into blue, it is called as a base. Next natural indicator is turmeric. Turmeric. Have you ever felt that turmeric is an indicator? We know that turmeric which we use in our kitchen and all, have you seen that we usually put it in the curries? If you have not seen, then go to the kitchen and ask your parents to show you the turmeric. Turmeric is a natural indicator. This we can easily do an experiment in the home itself. Just take a small white cloth or some, not a colored cloth, better you take a white or a light colored cloth. Put some turmeric solution and just make it completely in the towel, and just take a waste towel you can take, okay, and just put some soap solution in and just rub it. You can see that the turmeric color has been changed into a reddish brown color. It's changed into a reddish brown color. Do you know why? Because our soap is basic in nature. Our soap which we use in our body is basic in nature. So when a base reacts with turmeric, the yellow color of the turmeric changes into yellow changes into reddish brown in base. But this yellow have no color change in an acid. No color change in an acid. So I think you get it. That is turmeric. It changes its color from yellow to reddish brown in base, and the yellow color of turmeric does not undergo any change in an acid. So we can say turmeric is a natural indicator. Clear? Next one is cabbage. That also we are using in our kitchen. That is red cabbage. We have seen different color cabbages, yellow and red cabbages, purple. So we are using red cabbage as an indicator. We have to take the red cabbage solution, and if we put little bit lemon in that, lemon juice into, we know that in lemon there is acid. We know that in acidic in nature. So we are just adding little bit into this red cabbage solution. We are adding little bit acid. We can see that a reddish brown color will be produced. A reddish brown color. So the red cabbage solution, the red color changes into a reddish brown in an acid, and the red color change into a green color in a basic solution. In that you can add little bit soap solution. You can see that the red color solution, cabbage solution, gets turned into green color solution. So we got three natural indicators. That is, one is litmus paper, which we get from lichen plant. Second one is turmeric. Turmeric is a natural indicator which we can use as an acid indicator. It's indicator to identify an acid or a base. The yellow color of the turmeric color looks same in an acid, or it changes into reddish brown in basic solution. And the red cabbage solution is red in color. When we add acid to there, we can see the color of the solution changes from red to reddish brown in an acid and red to green in a base. Clear? So these are some of the natural. There are many. We are just discussing only these three natural indicators. Next comes our synthetic indicators. Next is synthetic indicators. Synthetic indicators. From the word itself, it's clear that something we are preparing. or some chemical substances which we are mixing to produce these type of indicators. So the first one which we are going to discuss is methyl orange. What is this? Methyl orange. It is also a synthetic indicator. So methyl orange, I think most of you have seen that in your laboratory. Methyl orange, it will be like a light yellow or like an orange color. So we are taking little, we have 2 test tubes and we don't know what all solutions are there. It is acid, maybe in one it will be basic solution. So in that I am adding 1 drop of methyl orange in both. In the first test tube, the color changes into red, and in the second, the color changes into yellow color. Then we can say that the solution which changes into red by adding methyl orange, it is acid, and the solution, the color changes into yellow, is called as base. If there is no color change, we can say that it is not an acid, not a base. It can be a neutral solution. There are many types of neutral solution, not an acid, not a base. So methyl orange is a synthetic indicator. So we can think that red shows a danger. So when we add methyl orange, if its color changes into reddish brown, we can say it is acid, and if it changes into yellow, we call it as a base. Clear? Next synthetic indicator is phenolphthalein. It's very familiar for, I think all knows that we have heard phenolphthalein. Commonly in the laboratory, we use phenolphthalein as an indicator. Commonly it is used, phenolphthalein is used in an indicator, as an indicator. So the same thing, we have 2 test tubes. One in which we don't know, both have the solutions A and B. I'm just adding phenolphthalein into both. In one, there is no color change. When we adding in a solution phenolphthalein, there is no color change, we can call it as an acid, and if the color of the substance changes into light pink color, then we can call it as base. So the phenolphthalein is an indicator to use to identify both acid and a base. If we add phenolphthalein to acid, it does not have any color change, and if we add phenolphthalein to a base, its color changes into a pink or a purple color. Clear? So these 2 are there are many. Methyl red is there, methyl orange, phenolphthalein. In the textbook, we just want to learn only 2, that is methyl orange and phenolphthalein. So these 2 are the synthetic indicators. The last comes the olfactory indicators. Olfactory means something which gives us a smell. Smelling. These are also, we can call it as a natural indicators. By the smell of the certain substances, we can identify whether that is an acid or a base. How we are going to identify using these olfactory substances, we will discuss. So olfactory indicators are the substances which changes their smell when they are added in an acid or a base. Olfactory indicators are those which change their smell when they are present in an acid or a base. How we can identify? A simple example, one olfactory indicator is our onion. That is onion. When we can take one onion and we just cut it to slices, and usually in all our home acetic acid will be there. Vinegar. You just ask your parents whether you have acetic acid in that. Just take some acetic acid and put onion into it. The smell of the onion does not change in that. So we can say that it's an acid. If we take a basic solution like sodium carbonate or sodium bicarbonate solution, and if we put onion into that, the smell of the onion we don't get the smell of the onion. So we can say that onion is an olfactory indicator. It does not have any change in the smell. We know that onion is having a pungent smell. So if it does not have any change in the smell when we put the onion into the acid, or the onion have a change, or we does not feel any smell when we put onion in a base. So onion is an olfactory indicator that it helps us to identify by using the smell. Second one is vanilla. We all know the smell of vanilla. Vanilla cake, vanilla essence. You have the smell of vanilla, isn't it? So we get vanilla essence from vanilla leaves. So we are taking little bit vanilla, and if we put vanilla into an acid, there will be no change in the smell of the vanilla. But we put the vanilla in a base, the smell of the vanilla vanishes, or we does not get the smell of vanilla. So we can say that vanilla and onion can be used as an olfactory indicators. It's also a natural indicator. Clear? So today we have discussed about indicators. Indicators are those which are used to identify whether it is an acid or a base. There are natural, synthetic and olfactory indicators. Natural indicators are those which are present in the nature. Example, turmeric, then litmus, then comes red cabbage, then comes synthetic indicators. Synthetic indicators, that is methyl orange, then phenolphthalein. And olfactory indicators, onion and vanilla. Turmeric changes its color from yellow to reddish brown in a base, and there is no color change in an acid. Litmus, blue litmus changes into red in acidic solution, and red litmus changes into blue in basic solution. And red cabbage solution. Red cabbage solution, having a red solution, we are taking the solution of that. If it changes into reddish brown, it is an acid, and if it changes into green, it is a base. Then synthetic methyl and phenolphthalein. Methyl orange changes its color into red in acidic solution and yellow in basic solution. Then comes phenolphthalein. Phenolphthalein is colorless in an acid, and it will be having a purple or a pink color in a basic solution. Then comes olfactory indicators. By smelling, we can identify whether the compound is an acid or a base. If we put onion in an acid, there is no change in the smell of an onion. But if we put the onion in a base, the smell of the onion vanishes. Vanilla, the same thing happens. If we put vanilla in an acid, there is no smell. If we put vanilla in a base, there will be no smell. But we put vanilla in an acid, we get the same smell of vanilla. Clear? So today with this we will come to the end of this topic. Tomorrow we will be discussing more on this acid. If you have any doubts, you can ask in the comment section. Okay, thank you.